Welcome back, everyone. The second part of our session. Now, I would like to introduce our second speaker for today, Prof. Dr. Antonella Motta. Prof. Dr. Antonella Motta is a full professor in the Department of Industrial Engineering, the chair for the Bio-Research Center and Biomedical Technologies in University of Trento, Italy. He is also the visiting professor of our university, UKM, Associate Director of Bioactive Resource for Innovative Clinical Application Research in Bangkok University, Thailand, and a member of the European Institute of Excellence in Tissue Engineering and Regenerative Medicine. Prof. Antonella has supervised about 25 bachelor students, 15 master students, and 45 PhD international students. Her research topic of interest include biopolymer-based scaffold design for regenerative medicine application, selection of new organism source, isolation process, instructive scaffold design and characterization, implantable device and matrices for tissue engineering application, in particular a 27 years experience on silk-based matrices for application in tissue engineering, such as bone, cartilage, and brain. Professor Antonella is also the editor-in-chief of the Journal of Biomaterial Science Polymer Edition, co-editor of a book on tissue engineering, scientific editorial board members in many international journals. She has published in more than 180 papers in various international journals and 14 book chapters. She was frequently being invited to speak in several international and national conferences. Without further ado, I would like to invite Prof. Dr. Antonella to deliver her talk on creating a sustainable research environment. Dr. Antonella.
Good morning. Let me first of all thanks to the organizing committee to invite me to give a talk at that conference at the Research Week. And they're giving me the possibility to share with you our approach to medicine with the concept of sustainability and circular economy. Let me start with the opinion of European community that is strongly supporting strategies able to follow the sustainability, green chemistry, and low environmental impact. This is not a very new concept, but what is relatively new is the application of this concept to the medicine. Medicine, where the idea is to combine material performances, human health and sustainability, and the core, the central part of the story are the use of natural materials, as I will show you during my talk. To reach this goal, the idea is to use materials derived from biological sources, from waste, industrial and food waste as well, and of course to follow biomimetic strategy in order to be more efficient in the application of this concept. This is the scheme of the production of new materials and devices for medicine, and what we are trying to combine is the quality control to have a faster application into the market, to the clinics, with sustainability, that is a concept just introduced in the previous slide. So the story starts from the selection of materials, raw materials. In our case, the idea is to use mainly biopolymers that should be engineered in order to have precision biopolymers able to be used in a different application, in particular considering the precision medicine, the personalized medicine. Then we have to move to the manufacturing and finally to the biological evaluation. What should be taken into consideration step by step? For example, in the use of polymers, if sustainability is our approach, we are considering first of all the use or the reuse as well. It's better to define reuse, industrial waste, food and, for example, textile industry. I will show you in few slides, to design reproducible protocols that can allow us to save materials, time, and cost, and to design manufacturing method. Try, for example, to reduce the temperature needed, the time of production, and of course, as a consequence, the cost. Finally, there are two more concepts that we use in the biopolymer selection and manufacturing as well. Is the selection of reactants, consider the impact on the environment, and eventually if these specific reactants should be used for the efficiency of the protocol, we should design other method to reuse and to recollect in order to avoid the impact, the negative impact on the environment. And finally, these are two concepts that Mother Nature knows very well: the recycle and the reuse. Manufacturing are used to produce then several types of medical devices, from implantable permanent devices to tissue engineering, artificial matrices, as well as the in vitro model for screening, personalized medicine, and testing. This construct then should be evaluated. This is another point we have to take into consideration. We have maybe to redesign, to reevaluate our protocols, consider that the starting material is a nature-derived polymers, so we have to design preclinical evaluation, and thanks to collaboration, external collaboration, to have a multidisciplinary team in collaboration with biomed companies in order to obtain, to try to move to the market sustainable products in medicine. So all of this is designed in order to reduce the time of production, reduce the cost, of course increasing the safety and the precision of our devices, as well as the specificity. What about the biomaterials? Of course, this is a general scheme that I use in my talks to demonstrate what should be done with the starting polymer. At the beginning, we have to select specific biopolymers as a chemical backbone, but these polymers should be designed to have precision biomaterials in order to design a platform that can be applied to several applications, but by starting from a small number of backbones and polymers, starting polymers that can be improved through functionalization in order to provide to the cells the suitable environment to promote tissue regeneration, for example. And then this depends on the final goal, if you want to have matrices that should be populated then by cells, or if cells should be entrapped during the processing. We can play with the cross-linking degree, we can play with the cross-linking molecules. We have the opportunity to use enzymes as cross-linkers. In this way, degradation of the material will be driven and controlled by cellular activities. That's the reason why natural derived polymers, once again, are very, very promising. We have a number of fabrication methods designed for these specific polymers. We can blend them with other natural derived or with the synthetic polymer when, for example, specific peculiar mechanical properties are required. We can play with the cross-linking. We can functionalize them in order to put into the structure the perfect language to instruct cells to promote what is needed for the therapeutic purposes. And of course, we can have also the possibility to include the releasing system. Just to give a brief idea what means reuse and the use of waste, for in this case from food, consider the crab that is used for human as a human food, but after that after the use as a food, we have a lot of waste. The waste are the carapax. Carapax is a chitin. Chitin is a very interesting polysaccharide that can be used as it is or can be deacetylated in order to obtain a chitosan. With the chitosan, we can obtain several medical devices. For example, we can use outside of the biomedical field the chitosan as a filters. For example, was proposed as a system for seawater purification, or can be used as addictive in the human nutrition, but it was seen to be able to treat, to have some positive impact in specific renal pathologies. Finally, we can have the use of the reuse of this polysaccharide in the textile industry. Some fibers can be obtained, and some textile with specific properties like anti-static or anti-acarose, just an example. But of course, what we are interested more is the reuse of this food waste in medicine, and several matrices are obtained. There are protocols to obtain a thermoresponsive hydrogel and so on. And also in this case is a polysaccharide. The polysaccharide has the several functional group that can be used to functionalize the polymer in order to have a precision material at the end of the story. Of course, we don't have just crabs. We have a number of sources, each one producing a specific chemistry. We have a protein like a collagen and silk. We have a polysaccharide, but we have also the mussel shell, for example, that can be used as starting material to obtain bioceramics. These polymers are obtained and the specific processing methods are designed in order to produce several strategies from cell entrapment to solid porous material, and several applications can be considered and approached. During my talk, I will focus my attention on silk polymers, but also in bioceramics. So we have several silk producers. Of course, we don't have just Bombyx mori. We have the wild silk, but we have other insects like bees that produce short silk fibers that they use in combination with the wax to produce a honeycomb to have better mechanical properties. And then we have well-known arthropods, but also mollusca produces silk. These filaments that are named byssus are made by the combination of silk and collagen together. But this mollusca can provide the shell and other food waste, of course, that can be used to produce a bioceramic. I will show you in the next slide what can be done and the value in the medicine. So what should be done when we want to start from food waste? Of course, not just mollusk shells, but also eggshell, cuttlefish bone, just example. These are material we are considering nowadays. And with the help of Professor Vincenzo Sglavo in our department, a specific processing method, flash sintering and cold sintering, were designed in order to produce bioceramics, reducing the temperature needed. Usually to obtain ceramics, sintering is done at degrees 700, 800 degrees, even higher temperature. But of course, we want to be more sustainable, reducing the energy involved. So new systems are designed trying to reduce as much as possible the temperature needed. Nowadays, we are almost close to room temperature. And this is quite important for a couple of reasons. Not just because the energy involved is lower with saver energy, so more sustainability, but also because in the muscle cells, for example, shells, there is not just inorganic phase, there is also organic phase that is very important for the biocompatibility. And if the process reduces the temperature, even the organic part can be saved, so improving the bioactivity of the final material. After the sintering, a powder is obtained, and this powder can be printed to obtain a scaffold for bone regeneration mainly, and thanks to the printability, several architecture can be obtained even with some gradient to approach applications such as, for example, the osteochondral defect. This is was done according to the Green Chemistry Pocket Guide that you can find in internet. There are 12 indications to be green. These are named the principle of green chemistry. So in this case, we can approach the number one, preventing waste. We can reuse. Less hazardous synthesis because we just use a low temperature to transform this organic bioceramic into the ceramic that can be also manufactured. Design for energy efficiency, reducing the temperature and the time of the properties, and reduce the derivatives because the process is quite clean. Just an idea on how we can apply this concept to the lab work, to the medicine. What is interesting, of course, is also the bioactivity, as I mentioned before. So first of all, the processability, design the process, reduce the energy involved, and reactant that can be can have a negative impact on the environment, but then we have to assess the bioactivity. So we produce some scaffold with the bioprinting by using bioceramics obtained from 3 different sources and compare with the commercial hydroxyapatite. As you can see in this case, this specific case, the bioceramics was combined with the synthetic polymer polycaprolactone and of course compared with the pure polycaprolactone. As you can see also in this time the possibility to use the printer, so we can bioprint three-dimensional structure, and then we culture cells. These were osteoblast cell line cultured up 7 days. As you can see by looking the green in the picture, you can see the difference between the pure synthetic polymer and the synthetic polymer in combination with the bioceramic. We did something like this by comparing with the commercial hydroxyapatite, and the results were very similar. The use of bioceramic is much more bioactive for bone cells because the presence of microelements that nature combine in the shell of the muscles that are very beneficial for the metabolism of the bone cells. Moving then to silk-based devices, what can be done to be more sustainable? So first of all, I would like to show you how is used. A number of applications are already considered nowadays. Several kinds of structure obtained, and several protocols were designed. And silk is not just a research level. We are doing more at this level, but we have already into the market a product silk-based, as you can see in this list, that are FDA approved and used for several applications, from tissue support to meniscus, anterior cruciate ligament regeneration. But what can be done to introduce sustainability in the use of silk in medicine? We have to go back to the multi-step process of the silk, and of course the silk is produced by the silkworm. This is the producer used mainly for silk in medicine. After that, the cocoon are collected, and the cocoon should be purified with a mechanism named the gummy that is used to remove the superficial water-soluble protein named sericin. And then this fibrin fiber should be used to fabricate by following, for example, the step of dissolution and then manufacturing final the storage of the protein-based device. What can be done to be sustainable? We should go step by step, considering the silkworm breeding, considering and controlling the environment of breeding from humidity, temperature, and the food we have to go to provide to this insect, controlling the life of the silk. We can control the quality of the silk. Then we should move to the gummy, for example. The silk fibers, we can use several protocols, but we have to take care about the chemical use, trying to define the minimum concentration to have the maximum impact, positive impact. We have to redesign protocols to reduce temperature, the time of treatment, and the volume of water used in order to be more to have a better economy into this step of the silk processing. Moving on, the device fabrication. These are the same concept as explained before. Also in this case, we have to reduce the negative impact on the environment, reducing time and hazardous reactants. Finally, the storage that should be possibly at room temperature, avoiding the use of -20 or -80 that can require a very high energy consuming. These concepts are recapitulated in a project that just started 1 year ago. This is funded by European community named Shaping Innovative Products for Sustainable Tissue and Engineering Strategies. It is our pleasure to have in the team Professor Fauzi Busra, that is taking care the production of matrices for skin regeneration, starting from collagen, mainly collagen. Let's go a little bit more more in detail into the protocols. For example, what can be done in collaboration with textile industry. You can see the multi-step recap into the slide. But what is the message of this slide is that each step can produce silk fibrous waste. It is a waste that can be short fibers, can be sericin into the gaming bath, can be obtained during the spinning, during the textile production, and considering 100% of the material at the beginning, what we can obtain is almost about 40% of the starting weight. This means that we have a 60% of waste that can be used, reused, and applied to different fields, from cosmetic already done to medicine. For example, sericin can be used as anti-cancer in specific application. What about the principle of green chemistry use? Of course, preventing waste, reducing the hazardous synthesis methods, design with the chemical that doesn't have a negative impact, design energy efficiency, for example, reducing time and temperature of the gummy, and reducing the derivatives, because derivatives are reused, recollected, reused for different applications. What about the use of protein from waste? That is a very nice example. This is a piece of work we did with a university in Colombia. That we try to recollect the sericin, the water-soluble protein that is coating the fibers into the cocoon, by using defective cocoon or normal cocoon or waste from the short fibers. That's to show you what it means. We have several opportunities. This is good, but this means that we should be very careful in the quality control, because we have a different waste of sericin, but depending on the waste source, we can obtain different chemistry. Just looking in some amino acid, as you can see, for example, in terms of alanine, we have different content moving from the Bombyx mori cocoon sericin isolated with the standard protocols, and compared with the defective cocoon, you can see is more than double the presence. If the chemistry change, means that the property will change, and we have different bioactivity. Is a more opportunity, but means quality control. And different chemistry means a different ability to assume a different conformation. For example, this is related to the fibroin isolated from different, sericin, isolated from different sources again. And because a different chemistry, if you look just at this histogram and consider, for example, the alpha helix conformation, depending on the source, the sericin can assume different crystalline degree, but also different conformation, increasing alpha helix or increasing random coil. And this means changing the hydrophobicity, hydrophilicity, but also the functionality and the impact on the cellular behavior. Moving then to the processing method, we are optimizing our method, again considering the possibility to reduce the time, the temperature, and the cost. This is just an example. This is a sintering of a solid fibroin designed by Professor Maniglio and Dr. Bucciarelli. They try to start from freeze-dry fibroin isolated from waste, freeze-dried, and the powder is treated in compression in presence of water with a very, very mild temperature, very close to the room temperature. And with this system, solid material are obtained that can be cut by laser, obtaining different shape. Of course, these are not needed for medicine, but just to show the tunability and the possibility to change the shape and to adapt the shape depending on the application. Even in this case, the biological activity was assessed and the cells were seeded. These were human mesenchymal stem cell. As you can see, in 10 days, the structure obtained by laser, by sintering and laser cut were completely populated by cells. You can see here they are differentiated in bone cells. Of course, sorry, in here you cannot see the differentiation, but just by shape. And then we did some gene expression to evaluate the ability of these cells to become bone cells and start to produce bone. Another concept is applied when the scaffold should be also functionalized with some drug release system. What is the concept of sustainability? To be very efficient, reducing the amount of the reactant used. What we have to do to reduce the drug? We have to load the scaffold. First of all, we have to identify the pathway we would like to activate, to be very specific. This is a nice example, piece of work done in collaboration with the Orthopedic Institute Rizzoli in Bologna and the Emory School in Atlanta, US. And the idea was to improve the ability of the bone to regenerate even in osteoporotic patient. And as a mechanism was identify the mechanism used by bone cells to produce endogenous sulfur that is needed for bone synthesis, in particular the pathway based on the beta-catenin. These people, the group of Rizzoli Institute, Dr. Grassi, identified this sulfur donor, GYY, and at the beginning this was used at systemic level, injecting to the osteoporotic animal, having beneficial effect, but with the need of daily administration of this donor. So we move to the tissue engineering. In this case, this donor was combined with a porous structure made by Silica fibrin, and we load with the sulfur donor. The releasing was assessed, and the sulfur donor GYY was released completely in 12 hours. Despite this short time of releasing, we perform in vitro test by culturing mesenchymal stem cells, human mesenchymal stem cells in dynamic condition up to 3 weeks. As you can see from these images, from this scheme, the presence of the GYY was able to improve the expression of osteogenic genes for 3 weeks, not just for 12 hours, and also the production of angiogenetic factor were activated and increased by the time. So what was interesting to observe that the 12-hour releasing of the GYY, the sulfur donor, was enough to trigger this mechanism based on the beta-catenin. So in 12 hours, the system was able to be reactivated, and then the cells start to do the job without the need of the releasing of more molecules. So this is a good example on how we can reduce drugs, how we can reduce the releasing time that is very beneficial for cost and for the time, even because we don't need to maintain active the molecules for a long time. But what is needed is to precisely identify the mechanism we want to reactivate, biomimetic approach, and then to release just the time to reactivate the mechanism. And this is a kind of sustainability. So just to summarize my talk, what is needed to move sustainability in the biomedical research is to design standardized protocols to be reproducible, to save time, and the validation protocol. We have to, into these protocols, we must be sustainable, we must be environmental friendly, and to move this concept into the protocols. We have to maybe to reevaluate, to redesign also the consensus testing, and to reach harmonization between regulatory bodies in the world, so we can have just one path for everyone. This can be done thanks to the international multidisciplinarity, collaborative network, and involving young researchers and teaching them how we can do research market-oriented from the beginning, not at the end of the story. This should be allowed now us to develop a sustainable practice in tissue engineering and biomedical technology. These again are the concepts in the project I already introduced at the beginning of my talk, Shift. And let me at the end introduce all our partner. We have two partners in Europe, University of Trent, of course, University of Birmingham, University of Minho, the Institute of 3B's Institute, but we have 5 partners in Asia. First of all, I would like to mention TERM and the Medical School and Professor Fauzi Busra that is proposing the use of collagen for wound healing treatments. Chulalongkorn University, where the research will be based mainly on silk, and and then Chonbuk University again with the collagen in South Korea, Mongolia, and they are exploring the possibility to use animal-derived proteins, animal hair like camel, horses, and cashmere for skin treatment applications. And to the other side, the 3Bs working mainly with the sea marine origin materials, and University of Birmingham are involved in the transnational biomedical research. With this, I would like to thank you for your kind attention. I hope that my translation is correct. And thanks again for giving me the opportunity to talk at this conference.